Welcome to everybody joining us today. Great to see everybody coming in. Just give everybody a couple of minutes, but welcome everybody if you're just joining us. I see the numbers are still going up a little bit, so yeah, we'll just wait for another minute or so. Okay, I think we'll maybe make a start then. So yes, thank you so much for everybody who can join us today on our RSC desktop seminar series. Today we're going to be focusing on drug development targeting cancer. And we're delighted to have some incredible speakers to present to you today. So first up, we're going to have Professor Mike Waring, based at Newcastle University. And he'll be focusing on approaches to the discovery of targeted covalent inhibitors. We'll open up the floor for some questions after that. And then our second speaker will be Professor Twan Lammers, based at RWTH Aachen University. And Professor Lammers will be focusing on tumor targeting using drug delivery systems. So just to go through some etiquette and code of conduct. So this webinar will be recorded. Attendees will be on mute throughout the webinar, but you can type any question that you may have into the question box. And then after each speaker has had a chance to finish their presentation, we can move to a question time. We do say that we would reserve the right to remove anybody who doesn't adhere to our kind of code of conduct and our policy around inclusion and diversity. So yeah, we agree as the Royal Society of Chemistry webinar series to foster equal participation, maintain privacy and confidentiality. We ask everybody to engage with kindness, respect, and please consider diverse cultural backgrounds. We will not tolerate any bullying, harassment, or discrimination, and we ask everybody to respect people's identities and experiences. We ask everyone to keep communication professional and contribute constructively, which I'm sure, of course, everybody will. So just a couple of slides just to introduce some journals that the Royal Society of Chemistry publishes. So first up, we have RSC Medicinal Chemistry, and one of our speakers today, Professor Mike Waring, is editor-in-chief of that journal. And that journal really focuses on medicinal chemistry and drug discovery research. We also have RSC Pharmaceutics, which is led by Professor Yvonne Perry, and we focus on drug delivery, formulation, and pharmaceutical technology. We also have Organic and Biomolecular Chemistry as a real broad organic biomolecular journal and publishes research across lots and lots of different areas, organic chemistry, supramolecular chemistry, physical organic, and the chemical biology space. And this journal is led by Professor Anthony Davis. And then finally, RSC Chemical Biology, which is led by Hiroaki Suga. And this journal focuses on research at the interface of chemistry and biology, looking at chemical tools to resolve biological questions. And it's of real interest and applies to researchers who want to explore mechanisms and manipulate biological processes at the molecular level. So just to introduce one of our newer journals, RSC Pharmaceutics, one of our speakers, Professor Twan Lammers, is an advisory board member for this journal, and I work as the deputy editor for this journal. And yeah, it's a gold open access journal focusing on formulation and design, dosage form, controlled release, and the kind of properties of drugs and their carriers. And just to flag that we are gold open access and all our APCs are waived currently until mid 2026. So an opportunity to jump on and take that offer at the moment if anybody is interested. Just to introduce our editorial board. So, as I mentioned, the journal is led by Professor Yvonne Perry, based at the University of Strathclyde. And of course, we have researchers based across the globe handling manuscripts in their relevant areas of expertise. So on to our first presentation then. So as I mentioned, Professor Mike Waring, based at Newcastle University, is going to be our first speaker. He's editor-in-chief of one of our journals, RSC Medicinal Chemistry. And yeah, just to introduce a little bit then about Mike. So Mike is Professor of Medicinal Chemistry at Newcastle University. Prior to that, he was a principal scientist in medicinal chemistry at AstraZeneca, and his research interests are in the discovery of cancer treatments and new approaches to medicinal chemistry. His current research focuses on the discovery of covalent drugs and understanding factors that influence their activity. And so just to introduce the talk today, so Professor Waring's talk is going to focus on covalent inhibitors, which have become a common approach to the discovery of new cancer therapies. And this talk will describe different approaches to their discovery, illustrated with SAR studies from Professor Waring's lab. Factors influencing covalent binding and their optimization and methods for screening will also be discussed. So thank you so much for being with us today, and over to you, Mike.
Okay, let me share my screen. Okay, just to check this is all working okay. We did have a test, but
yes, looks good to
okay,
me.
brilliant. Thank you. So yeah, thank you all for joining. It's a great pleasure to be able to tell you about some of this work. Yeah, so what I'm going to talk about is a few different case studies. So I've got 4 different stories that relate to kind of illustrative work that we've done in this kind of area. So I was going to go through those and try to bring out what I think are some of the general principles around covalent drug discovery that I think are considerations, in a lot of cases, unanswered questions, I think, for how we might develop or discover covalent inhibitors in future. So I'm not going to suggest that these are all the answers. These are not really telling anyone how to do it, because I think they're just some discoveries that we've made along the way that are things to consider if you work in this kind of area. So I'll start with a little bit of an introduction, and I'll go through these the case studies that I mentioned. So for those of you who are less familiar with the area, this is a class of drugs that, particularly in the cancer field, have become undergone a bit of a rebirth over the last sort of 15 years or so, mostly catalyzed by the discovery of a few prominent inhibitors that have become cancer medicines. And I've just shown 3 of them on this slide here. So ibrutinib, which is an irreversible BTK inhibitor, osimertinib, which I'll say a little bit more about, in fact, become the kind of central theme in the talk, actually, which is a compound that we developed at AstraZeneca and is now a very successful treatment for EGFR resistant lung cancer. And then finally, more recently, this compound sotorasib, which is a KRAS G12C inhibitor. And you can see they all have common features, so that they all work covalently, and they all work by having this acrylamide residue that is the trap for a cysteine that forms a covalent bond with the cysteine. So in a little bit more detail conceptually in terms of discovery approaches, really the approaches to discover covalent inhibitors have been broken down into 2 different kind of categories that have become known as reversible first or electrophile first approaches. And really they differ in the sense that if you have a reversible first, as the name suggests, you start by discovering a non-covalent inhibitor that binds to the target, and then you look for some kind of proximal nucleophilic residue, which is usually cysteine, and certainly this talk will be confined entirely to cysteine as a nucleophilic residue in the protein. And then you attach onto your irreversible lead some kind of electrophilic trap that will then react with the cysteine and form a covalent bond. So this requires you to already discovered an inhibitor of the enzyme that already binds, or a ligand for the enzyme at least, that already binds and has a proximity to the nucleophilic residue that you want to target. The alternative is to do things the opposite way around, which is called electrophile first, where you screen a series of electrophiles, look for something that reacts with the nucleophilic residue on the protein, and then you try to grow that group to develop the inhibitor. Okay. And going back to those three examples I've just shown you, so ibrutinib and osimertinib were both discovered via the top approach, and sotorasib is, at least in part, an example of the bottom. And I just want to go a little bit more into the background to this and talk about the discovery of this compound, osimertinib. So this is a story that I swore every time I tell this story, I'll not tell it again because it's becoming old hat. But I would like to just go through this now, both to set the scene and to bring out some of the advantages of this approach as a way to target cancer in particular, but also to throw up some of the questions, to lead into some of the questions that I think we've been trying to answer in some of our research more latterly. So the story, a background to this story is that the target is this protein that's shown on the screen here, which is the kinase domain of EGFR, which is a receptor tyrosine kinase that promotes oncogenic signaling in lung cancer by autophosphorylation of two residues upon binding its activating ligand. The background biology to that doesn't really matter for the purposes of the story that I'm going to tell you. And it's been a target in lung cancer for a long time, and this is one of the first inhibitors one of the first clinical inhibitors of the protein is shown here, gefitinib, which is a reversible inhibitor. And the story around this, the discovery of osimertinib, really relates to these mutations that are shown in the slide here. So the first thing to say is that EGFR becomes activated in a certain subset of lung cancer by these two mutations here, either a point mutation in residue 858 that turns from leucine to arginine, or the deletion of this series of residues here that correspond to exon 19. And both of those things activate the protein towards increased signaling and therefore oncogenesis. What they also do is diminish the affinity of the receptor for its natural substrate ATP, which means that you can get some selectivity for the mutant forms over the wild type with competitive inhibitors, because you pay less of a penalty to compete off the ATP for the mutant form than you do for the wild type. And that's really important because wild type EGFR is associated with some pretty severe toxicities. And this is a successful treatment, but you get resistance. And one of the primary resistance mechanisms that happens in about half of the cases is the mutation of this residue here, T790, that becomes a methionine. So those of you who work in the kinase space, you'll recognize this is the gatekeeper residue, which has a profound effect on the types of inhibitors that bind. And the mutation of this residue to methionine really diminishes the affinity of non-covalent inhibitors like gefitinib for the receptor. But what it also does is increases the affinity of the receptor for ATP again. So you have that problem of competition. So we set out to try to develop inhibitors that were selective for this double mutant form that contains methionine at 790 as a way of treating first-generation resistant lung cancers. And we started with this compound here. So this is definitely an example of a reversible first approach. We were fortunate enough to already have a series of compounds that were active against other methionine gatekeeper kinases that we could test. And this compound here came actually from the IGF program. And straight out of screening, we were able to identify this compound as a really potent inhibitor of the of EGFR. So I should say, just in case, for absolute clarity, this presentation all uses PIC50 values. Okay, so if you're not familiar with that, these are numbers on a log scale where the higher number is the better. So this difference here pertains to 2 orders of magnitude difference in affinity. And what I've shown here is the potency of the compound versus the enzyme versus its activity in a cell-based assay that has doubly mutated EGFR. And you can see there's a problem here because the 2 numbers don't tie up. So even though this compound has a PIC50 of 8, so 10 nanomolar inhibitor, when you test it in a cell assay, it's 2 orders of magnitude less potent. And the reason for that really is because of this problem with the ATP affinity. And because the ATP affinity of the double mutant is higher, it becomes very hard to get potent compounds in a cellular setting if they bind reversibly. So what we did as a solution to this was to try to convert them into covalent inhibitors. And you can see here from the crystal structure of the compound bound to the receptor, there's a convenient cysteine that's located just in this region here that you could potentially target. And within very few compounds, we basically put the standard acrylamide trap for the cysteine onto this end of the molecule, and we can find compounds like this that now solve that problem. So at least in terms of the enzyme potency, don't seem to change very much, but they have much less of a drop between the enzyme and the cell because we now have non-equilibrium binding through formation of this covalent bond. So that was got us into the area of covalent inhibitors through necessity. And we were able to, cut a long story short, we were able to develop this compound into what became this structure here, which became osimertinib. Basically, the rest of the optimization story here is a story of fixing things like pharmacokinetics. You can see here this has no bioavailability. We could manage to solve that by making these changes that control the lipophilicity. And then it ultimately ended up with this compound here. The changes that are from here to here that I won't go into really are about trying to get the right selectivity. So the right selectivity profile for EGFR mutants and selectivity away from other kinases and hERG and all those other things. But you can see what's happened during this optimization process. We've retained this acrylamide trap that's reacting with the cysteine, and the overall shapes of the molecules have not changed very much. So this became a very successful drug. Last year, actually sold $6.7 billion, and a whole load of patients that have really benefited from that. But it was also a very quick discovery story. So the compound was first approved by the FDA in November 2015, which is only 6 years since that initial screen that found that compound I just told you about. So the whole discovery and the development was done in that short time scale. And so I tell you that story to lead into some of the questions and the points that you can take from that. So the first one, and the one that everyone asked at the time, was why was discovery so fast and why isn't it always like that? And of course, the speed you get to an endpoint is very much defined by the point at which you start. And we were fortunate to start with some really potent compounds and also with a decent amount of knowledge about how we could convert these reversible compounds to covalent drugs. And so that kind of prompts the question about what you do if you didn't have that kind of start point. So is a covalent first approach feasible? And since then, the stories around compounds like sotorasib have emerged that show that it is feasible. But I think present some challenges that I'll go into, in particular around how do you introduce potency into something that's already that gets all its affinity from a covalent interaction. So that leads you into the possibility of developing covalent fragment libraries and covalent lead-like libraries that you might screen up front to find covalent inhibitors. And I think my experience really would say that I think always, if you can find reversible compounds first, it's usually better to do that. So it's usually easier to convert a potent reversible ligand into a covalent drug if you have structural information and you have potent compounds, than it is to start de novo from designing covalent inhibitors from scratch. And I'll go into a few examples of that in a minute. The other question, of course, is do they present a development risk? So when we did this, we did it reasonably reluctantly. This is against the background of people worrying about things like reactive metabolites and things like that. So putting inherently reactive molecules into the body presents some kind of risk. And it's very hard to de-risk, actually. So even despite the successful stories, I think it still presents a question about will these compounds cause non-specific modifications of proteins, and will they lead to toxicities? And I think it's very hard to assess that up front, and you carry that risk through development. And I think you can get away with that if you develop molecules in cancer. Maybe it's less easy to get away with it in other areas. And in particular, in the case of EGFR, it was possible because really what we were worried about was wild-type EGFR tox, which was always going to dominate the kind of adverse effect landscape. And so maybe any risks presented through having covalent inhibitors were kind of hidden. But it does prompt this idea that if you have a new project and you have a structure and you see a cysteine in the active site, someone's always going to suggest now that you try to develop a covalent inhibitor. And I'm not sure that that's always a good thing to do. If you can get your affinity elsewhere and get reversible compounds, so maybe that's still like if there's no need to go after a cysteine, then maybe you shouldn't. That's maybe a question for discussion. And the other question is, can you always use the acrylamides? So certainly at the time when we worked on this program, generally they were the compounds that had gone through into development, and ones that were we thought for that reason presented less of a risk. And if we took a novel covalent warhead, that would presumably present more risk around development that again will be difficult to de-risk. But I think you can't always use acrylamides in this setting. I'll show you another example of that in a minute. And then the other question is, how do you drive the chemistry? So you will notice in the story I told you that we just use standard IC50 values against the enzyme and then cellular PD and efficacy readouts to assess whether the compounds were getting more potent. So we didn't do any Ki-Kinact kinetic measurements during the course of the project. And indeed, the Ki-Kinact values for osimertinib were only published here 5 years after the drug was approved, actually. So try as we might, we didn't get that information during the discovery program. It didn't influence the discovery of the drug. And the corollary of that, of course, is you didn't need to do these things to discover the compound. And I say that because there is a groundswell of opinion now that suggests that if you're doing covalent drug discovery, you need to be measuring Ki-Kinact values to drive your SAR. And I don't think that is necessarily always practical. It's certainly very expensive and time consuming, and it's not required. It's very nice if you have it, but then there's a question about how you use that information. And I say that because there is obviously a very standard understanding that in a purist sense, IC50 values against for covalent inhibitors are meaningless because they're time dependent, and that you really should get these parameters measured. But actually, I thoroughly believe that if you configure your assays in the right way, the IC50 values that you get from standard biochemical assays are not meaningless in the sense that they can still be used to drive chemistry, and they can be interpreted so long as you understand the limitations as to what they're telling you. And the other thing is that we didn't really have any X-ray structures. I showed a couple I showed one structure that showed that we had a covalent bond formation in the initial lead, but we didn't really use structural information to further develop the compounds. And indeed, another group solved the structure of osimertinib bound to EGFR later and published it. And in their X-ray structure, they don't see the formation of a covalent bond. You can see this here. The binding mode as you expect, the acrylamide is proximal to the cysteine, but there's no density for a covalent bond formed between them. And that's kind of intriguing. All day long, this is a covalent inhibitor, and yet you don't see it in the X-ray structure, which asks maybe a few other questions. So I'll just run through a couple of examples now of these case studies that I mentioned. So this one is not related to EGFR, but it relates to this question about whether or not you need whether you can use an acrylamide in all settings. And this relates to another kinase called Nick, where we have these compounds here that were actually literature compounds that bind in a way that's near to a cysteine. And we tried to make covalent ligands of them by pruning these inhibitors back to structures like this, and then introducing acrylamides. And you can see if you do that, you see drops in affinity and no suggestion that there's covalency. And the reason we think for that is really that the acrylamide itself is a polar group and it's not tolerated in this lipophilic pocket at the background. So what we did was to use these alkynyl heterocycles as alternative covalent traps that wouldn't be quite so polar. And you can see when we do that, we can at least get retained affinity in the biochemical assays for certain types of heterocycles in the middle, and the terminal alkyne here acts as the covalent warhead. And you can see it does depend on the position of the alkyne. Other isomers of this pyrimidine are not potent. You have to get this bit right. And we were able to show definitely that this is a covalent inhibitor by mass spec. You can see a time course for covalent labeling. You can see the adduct formed, and if you do the triptych digest, you can show that it's the cysteine that we intended that is labeled. And this just shows a dot structure that explains the regiochemistry around this. So the pyrimidine nitrogen we think is important in this kind of hydrogen bonding interaction to this lysine that forms some non-covalent interaction, but also activates the alkyne towards reaction with the cysteine. Okay, so back to EGFR. The other thing that we've done almost incidentally, so we did this study really because we were interested in making these compounds for other reasons, which I won't go into. But we were interested in making analogues of poziotinib that we were going to derivatize further, but we happened to test them all in EGFR and found some interesting SAR. So what we'd done here was to take this compound, which is a known EGFR inhibitor, poziotinib, which at least at the time the SAR for this was not published, but we had to make some different analogues because we're interested in different geometries and different affinities of these compounds. And so we made a series of analogues that varied this linking group here, different cyclic amines. You can see there's not big changes. We've just gone from 6 to 5 to 4-membered rings. And we changed this aniline that binds in the back pocket through different sort of halogenation patterns. And we tested all these compounds using this assay, which is a probe displacement assay that uses a TR-FRET between this fluorescent probe and terbium labeled EGFR. You can see here, this is the SAR that we got from making all those different pairs. And you can see the question that we were asking here is, can you interpret SAR from this assay? And you can see that if you look at the assay, the potency for the back pocket that's shown for these groups here, you can see differing potencies. You can see an order of magnitude difference between, for instance, this aniline and this one. It disappears somewhat when you have the more reactive propargyl amide. Okay, so I think that's an interesting thing, that you can see the SAR when the reactivity is lower, probably because the assay that we have is configured to detect differences in compounds of this level of reactivity. And if the reactivity goes up, those subtle differences disappear. And you can see the same thing for the linking group between the warhead and the core. Okay. Just to emphasize this point a little bit more, you can see this in the matched pair plot. If you look at the difference between the acrylamides and the propiolamides, you can see here that there's a much bigger spread in the acrylamides, and they tend to condense down into a much smaller spread when you look at the more reactive cores. Okay. The other thing that's interesting here is that if you drill a bit more carefully into the SAR for this linking group, we saw a big difference between these two compounds here in the acrylamide series. And you can see there's about an order of magnitude difference in the potencies between them. And they only differ a bit by the fact that they're two different enantiomers. And so we were interested in why that was. If you look at the mass spec labeling of the protein, you can see they're both definitely covalent, but there's a difference in the degree of labeling that you see by mass spec. So you can see this more potent one labels the compound a little bit more than the other one. And that difference in potency that we detect in this enzyme assay translates into pharmacology. So this shows the inhibition of cellular phospho-EGFR. You see the more potent compound in light blue here is more potent than in the phospho inhibition than the less potent one, and that translates also into growth inhibition. And you can see it translates more or less quantitatively. So these values that we got from here correspond quite nicely to the growth inhibitory potencies that we see in the cell assay. This shows the crystal structures, and we see the same thing. So the less potent compound doesn't show a covalent bond in the crystal structure, even though we see it in the mass spec, and the more potent one does. And we could rationalize this by a change in the kind of conformational energies of the compounds that need to get into the position to form the covalent bond. So the encounter complexes need to rearrange in a way that presents the acrylamide in the right position. And you pay a greater penalty for rotation about this bond here in one enantiomer than you do from the other. This is also apparent in the Ki-K-enact data when you measure it. So just to show that this does translate, you can see the more potent compound has a K-enact over Ki that's much greater than the less active compound. And it seems to be really driven by a change in Ki rather than K-enact if you drill into the differences. And so that suggests it's really something to do with the geometries, as I said, and the way that's accommodated. And just to back that up, if you don't have covalent inhibitors and you look at just the simple acetamides, there's not that much difference in potency between them. In fact, against the double mutant, they're pretty much the same. So it must be driven really by the formation of the covalent bond. And so then one final story that I wanted to tell you before we finish, I think we've just about got time, is a story that we've done similarly coming back to osimertinib that relates to covalent fragments. And this is really answering this question about electrophile first, what do you do if you don't have an established start point? And what we were interested in was to understand what would this discovery story of this compound look like if we'd have done it the other way around? So can we deconstruct this optimized compound into its component fragments and then make variations of those fragments and see what the SAR would have looked like? So this is a little bit of an academic project. We're only looking really for what that SAR would have been on a compound that's already discovered, but we thought that would be an interesting kind of experiment to do. So what we did was to deconstruct it into components where we remove the head group, and then we look at variations in fragments that have either the warhead plus this ring here, or the warhead plus this ring plus this one. So we made these compounds here that have variations of different the components of the structure, but also sort of simple analogs of them. So you can see here we've made analogs that either do or don't have the methoxy or move the methoxy around. We've made heterocyclic versions, and in this case we've moved variations of this ring where we've moved the nitrogens around this ring, and the ones that highlighted have the key interaction, which is the hinge binding region for the interaction to EGFR that we'd expect to be more potent, and some of the others don't. And then for good measure, we made this compound here that won't really say any more about that. And you can see if you look at the data for this, again, using the same assays and using mass spec and this displacement assay that I mentioned, if you start with the single ring fragments, you can't really differentiate anything between them. So it's not really clear why you would be able to optimize from these kind of simple fragments. You can see there isn't very much in the binding assay. If you look at these compounds here, you see signals basically because the reactivity's been jacked up. So you don't really get any SAR from that. Potentially you can see things in the mass spec, so you can see modifications of some of the compounds and others you can't. And maybe that means that if you start with very small fragments, you're really reliant on mass spec to detect whether anything's binding. You can't see it in these displacement assays. And to preempt a question, you can't see it in KIK INAC determinations either within the concentration range that we can test. If you go to the 2-ring fragments, that's not the case. So once you do this, you can see potencies that come on scale in this assay. In this case, you see single adduct formation in all of the mass spec data. And you can actually start to tease out the SAR. So this may not be immediately apparent from here, but just to crystallize that more visually, this is a plot of the compounds that contain that hinge binding group in the right geometry versus those that don't. And you can see those that do are clearly different to the ones that aren't in terms of their potency in this displacement assay. So at this level of this, we can start to see non-covalent SAR, and we don't just see things that are dominated by the covalent binding. So what we conclude from that, I think, is that you need, if you're going to start making covalent fragments, you need them to have this sort of level of complexity, so 2 rings of the order of 10 plus heavy atoms before they can start to be useful. Okay, in the interest of time, I'll just skip over that and just summarize. So what I hope this has covered a little bit, and as I warned you at the start, I think this asks more questions than it answers. That there are many factors that influence covalent binding. So electronics of the cysteine, hydrophobic environment of the cysteine, and the geometries of the inhibitors are all important. It's not just as straightforward as attaching an acrylamide to an existing ligand. If you want to determine SAR to drive chemistry, then I think hopefully this shows that IC50 values can show SAR, provided you look for compounds that are of similar reactivity and you can configure your assays appropriately. And you certainly, if you work with compounds that reactivity is too high, that SAR tends to disappear. It is definitely difficult to interpret SAR for covalent fragments. And as I said, we think that we need things that have fragment complexities of a level of 50 normal heavy atoms before you can start to see anything. And then the final thing that came out from pulling this together is a question that I think is, how do you prove a covalent mechanism? So I've shown you some examples here where you can see covalency by mass spec, but you can't see it by X-ray, including for optimized compounds. And sometimes maybe we can see, we have shown an example there where you can see mass spec labeling for a compound which you know is suboptimal in terms of its covalent interactions with that inactive enantiomer, and you don't see covalency in the crystal structures either. And you do probably pick it up in cellular pharmacology. I think maybe that's one of the things not to forget. So I will stop there. Just want to finish by acknowledging a few people. This is a picture of our drug discovery group taken recently. And without naming any names, I'd just like to thank the team at AstraZeneca that I work with on osimertinib. I've picked out a few people here, but obviously this is a much wider team who have all contributed. And similarly, my group at Newcastle that have driven a lot of this work, and I've highlighted a few kind of key players on the stories that I've told you about. And of course, I should thank this group of people that funded all of the work that I've included in the presentation. And thank you for listening.
Wonderful. Wow, really inspiring. Thank you so much for that, Mike. Yeah, we have 2 questions, maybe one extra one.
Just 1 second, I've lost my audio. It'll take me a minute to get it back.
Sure, yeah.
Battery 90%.
Okay, is that better?
Yeah, we can.
I got you.
Hear you.
Okay.
Yeah, okay.
My speaker power's down if no one else speaks. That's probably.
Yeah, just a couple of quick questions then. So Moad has asked, how can we differentiate the agonist and antagonist agents on the basis of covalent interactions?
Can you do that? I don't know. I guess all of our work has been on enzymes, so we've always worked on inhibitors. We've never worked on covalent activators of anything, receptors or otherwise. So I don't know. I guess there's no reason to think that an agonist would have different considerations. I don't think, but I'm making this up really.
No worries. Nope, that's wonderful. Thank you. And then we'll probably just have to ask one more in the interest of time. But Manassa has said, "Hi, Mike. Thanks for the interesting talk. I was wondering what your opinion was in developing acrylamide-containing covalent inhibitors for infectious diseases like malaria, especially if there is a non-catalytic cysteine in an allosteric pocket that confers selectivity over human proteins."
Yeah, I mean that is another setting. I think in infectious disease is obviously another setting where you can get away with covalency. You know, you'll be giving compounds acutely, presumably. So some of the long-term effects that you might worry about around reactivity are probably not so important. In the advantage that you have in the sounds like in the case that you're describing is that you can hit the target very hard with a covalent inhibitor. And you should be able to get prolonged residence time, which would have advantages for that kind of setting. Obviously, there's a wide precedent in infection for covalent inhibitors that go all the way back to penicillins, which are obviously a big class of covalent inhibitors that were discovered in the first time around when they were in fashion. I think the other advantage that you touch on, of course, is that it's a good way of getting selectivity. If you've got a unique cysteine in your target that's not in an anti-target or a human homologue, then it's a really good way of exploiting that kind of selectivity where you may not be able to get it in another way. The disadvantage you might find in this setting, and it's obviously something that we see with in cancer as well, is that there's an obvious resistance mechanism. So if the cysteine is not catalytic, there's an escape mechanism by which that residue can just mutate to something else, and then your inhibitors won't work anymore. That definitely happens. It happens with ibrutinib and osimertinib over time, and so you get reasonably durable responses, but eventually the cysteine to serine mutation is a common one that causes resistance. So, and I would guess in malaria that would be something that would come up more quickly.
Wonderful. Okay. Thank you so much. Okay, yeah. So that's great. So we'll move on to our second speaker of the day then. So Professor Lammers, based at RWTH Aachen University. And Professor Lammers will be talking about tumor targeting using drug delivery systems. A little bit of background. So Professor Lammers started the Nanomedicine and Theranostics group at the same institute around 2009. And in 2014, he was promoted to full professor of medicine, and his group aims to individualize and improve disease treatment by combining drug targeting with imaging. He's received many awards, ERC Starting, Consolidator and Proof of Concept grants, the CRS Young Investigator Award, and many other wonderful awards there. And since 2019, he is included in the Clarivate Analytics list of highly cited researchers. So I will pass over to you, Tuan, in that instance, that you can introduce your talk.
Thank you, Sarah. I'm trying to share. Now I can. Here we go.
Perfect. Yeah.
You can see my screen well?
Yep, that looks good. Yep.
Perfect. Let me grab a pointer. Yes, thank you. So as some of the attendees may have noticed, we switched orders. I was supposed to go first, but I guess, you know, a mutual agreement, we decided that it would be nice for Mike to go first, because I think molecular design is where things come first. And if there would be time enough, I would have asked the question, like, how did you consider things like oral absorption for these compounds, or how do you consider biodistribution, right? So how do these all nicely designed molecular compounds, how do they get to the right site where we need them to elicit their specific conjugated or non-conjugated, reversible, non-reversible effects? And that's sort of where I'm starting. So what we try to work on is try to understand the pathophysiology of tumors, to develop tools and technologies to either image the process, guide the process, develop biomarkers, and to implement those to promote clinical translation of anti-cancer drugs. Those can be based on drug delivery systems, on nanomedicine, but they don't have to. I would guess the first part of the 20 minutes that I was allocated, I will talk a little bit about the basic principles and some of the problems that we've had in the field of tumor-targeted drug delivery and nanomedicine design. And in the second part, I'll propose two different, solutions to how to deal with the issues that, in my or our opinion, are prohibiting more efficient, clinical efficacy of cancer nanomedicine formulations. So when we think about the tumor-targeted drug delivery process, this is very inefficient. And I think imaging is a nice way to understand how things work. If we look at the biodistribution of a carbon-11 labeled chemotherapy agent, many of the agents that Professor Waring was talking about they're related to kinase inhibitors. Many of those are designed to be not only good molecular inhibitors, but also orally available. In the old days, like 10, 20, 30 years ago, when people were mostly working with traditional chemotherapeutic toxins, everything was going intravenously. One of the key advantages of kinase inhibitors or other types of novel medications is that they are orally available. But in both cases, what is actually known for many small molecule compounds with very nice affinity constants, etc., is that they do not show a good biodistribution, which on the one side means they don't achieve enough or high enough concentrations in tumors. At the same side, they do accumulate at lesions where they cause a lot of side effects. And what we do with drug delivery is we try to balance those two components. So what you see here is actually a PET imaging, positron emission tomography patient with metastatic mesothelioma. This is a thoracical disease. So in the lung chest area of the patient, you would have a lot of tumors. You actually have a lot of tumors. And what you're seeing here is the distribution of a chemotherapy drug that is used in mesothelioma, labeled with a radionuclide labeled version of the drug, right? Carbon-11. This is the needle where the drug goes in. This is the heart. So the heart distributes the drug all over the body. Within the first 5 minutes, you already see a lot of the drug, probably 80%, 60% already in the liver. The percentage of the drug that is in the liver will not be able to escape back from the liver into the bloodstream to reach the tumor. So typically it goes out, and you see that over time, it goes out into the hepatobiliary system or into the stool. This is actually the urinary excretion pathway, which happens via the kidneys. So drugs are excreted very fast from the body. They're degraded quite fast within the bloodstream or within the liver. They don't reach their pathological sites. This is another example, doxorubicin, very old traditional drug, not as fancy as some of the new kinase inhibitors we have for specific molecular targets, but the most widely used and arguably also the most, let's say, broadly applicable anti-cancer drug. So it's used for 15 plus cancers. It's a very potent drug. It can only be given 6 times to patients because of the fact that it has a very poor biodistribution. So it accumulates in many healthy tissues, not very well in tumors. This is mouse data. This is concentration over time. You see a pretty good or, bad, good, high accumulation in the heart, which results from the intravenous injection of doxorubicin into the bloodstream via the tail vein in mice, which eventually results in cardiomyopathy, damage to the beating heart muscle cells, which is the dose-limiting side effect for free doxorubicin, and which limits the total number of administrations to 6. So you will never, in a clinical setting, find somebody that administers doxorubicin, even though it's working, more than 6 times. So we need to deal with this suboptimal balance between on-target and off-target efficacy or delivery. And that's what we can do with drug delivery systems, right? In the last 2 decades, many fancy names were envisaged for dealing with those formulations. Nanomedicine is sort of the overarching terminology. We have liposomes, we have polymer conjugates, we have micelles, antibody drug conjugates, dendrimers, all sorts of nanoparticles, various different types. Some of the ones plotted on the top more relevant than the others. We have multiple different targeting opportunities, from passive targeting, about which I will be talking most of my talk today, to active targeting to trigger drug release, with the overall goal of achieving basically what you see here in green. So on the one side, to protect intravenously or orally administered drug molecules from the body, to prevent them from being degraded, from excreted, and from for instance, not being absorbed properly. So we basically help the drug to get into the body in the right way to achieve sufficiently high concentrations. At the same time, drug delivery systems protect the body from the drug. So they prevent the drug from going to sites where we do not want to have it, which is the heart in the case of doxorubicin, or the central nervous or the peripheral nervous system in the case of taxanes, the kidney in the case of platinates, etc. So we basically balance both arms that influence what we refer to as the therapeutic index, which is the balance between efficacy on the one side and toxicity on the other side. In the drug delivery field, historically, people have always been talking about the EPR effect. EPR stands for Enhanced Permeability and Retention Effect. Was first coined almost 40 years ago by Professor Hiroshi Maeda from Japan, who was nominated for the Nobel Prize multiple times for coining, for inventing, discovering the EPR effect. Professor Maeda passed away a couple of years ago, so he will unfortunately never get the Nobel Prize again, which is, I guess, partially good, because many of the initial basics that he defined or uncovered are now sort of refined or disproven. But the bottom line feature is we do see that long circulating macromolecules, he worked with proteins, with polymers, not with traditional nanoparticles, not with liposomes, but he did see that nano formulations accumulate in tumors. And you see that here. So in black, you have the blood vessels running through a tumor. And in white, you see fluorescent macromolecules leaving the vasculature specifically within tumors. This is because the blood vessels in tumors are more leaky, more permeable than the majority of healthy tissues. Obviously, the liver and the spleen, etc., they also have an open vasculature. But for instance, the blood vessels in the heart, they don't have fenestrae or tight junctions that are large enough for nanoformulations to pass across. So that gives us an opportunity to actually use this local leakiness to accumulate nanoparticles in tumors. And what Professor Maeda hypothesized is that also lymphatic drainage within tumors helps to make nanoparticles or macromolecules accumulate within cancerous lesions. And I think this is probably the most important point that we now understand is not correct, because we now know that it's mostly macrophages in tumors that are responsible for this R component, the retention component of the EPR effect. And then sort of the bottom line thinking is that the longer something circulates, like antibodies do this fantastically, right? Evolution developed IgG antibodies to circulate very, very long periods of time to maximize the chances that whenever you have a local inflammation, which tumors in the end are, right? They're persisting wounds, they're not non-healing wounds, to exploit local vascular leakiness to eventually accumulate. And this concept we can use for efficient nanoparticle delivery to tumors. So multiple people have shown that this concept holds up in patients. This is an example from Alberto Gabizon. This is the very first patient ever dosed, not on the right, but on the left, with either a nano drug or a free drug. The free drug, free doxorubicin, as you'll see, is excreted from the blood within minutes. So the half-life time is 5 minutes. That means within 1 hour, it's below 1%. It will not go to the tumor. If we put the drug in a nanoparticle, we see a half-life time of 2 to 3 days, which means that if this EPR, this vascular leakiness, helps to accumulate drugs in tumors, we can actually use that to improve delivery. And that's what you see here on the right for this ideal case scenario. It's a Kaposi sarcoma patient. In Kaposi sarcoma, patients have an underlying HIV infection, so they have very high underlying inflammation, systemic inflammation, which leads to very bloody, very vascularized tumors with a lot of immune cells. And in all of those lesions, this is the primary tumor of the patient here in the leg. This is a metastasis in the foot, which you see here. And in black, color-coded here, you see accumulation of nanoparticles radiolabeled in tumors. So they do reach primary tumor metastases, and liver and spleen, which both also have a lot of blood vessels and also an open vasculature and also a lot of macrophages. So the biodistribution in a way for nanoparticles is defined to highly vascularized tissues with a leaky vasculature and a lot of macrophages. So that notion we can try to exploit for better drug delivery purposes. If we look at what we have in the clinic at this point in time, by the strictest of definitions, we have some 20 maybe 23, 24 products on the market. Doxil, liposomal doxorubicin, the example I showed you before, already has a couple of generics. So India and Taiwan, they all have their own type of liposomal doxorubicin. We have albumin-based or protein-based nanoparticles, condensates with albumin and paclitaxel. We have inorganic nanoparticles that are typically injected into tumors, so they don't exploit the EPR effect or injected locally, but they're approved for the treatment of cancer in the clinical situation. And we even have formulations in which we optimize the ratio of two different drugs to achieve synergistic drug interaction, in which one drug effect plus the other drug effect is more than two. So you really synergize the pharmacology to cluster the effects by enabling delivery to the same site at the same ratio via a nanoparticle to achieve, for instance, better therapeutic responses, in this case for this product Vyxeos in leukemia, but with many other formulations now in preclinical and clinical evaluation. The problems, all right, these are the principles, and we have a good number of products on the market. But if you look at the literature, there are so many papers on cancer nanodrugs, and everybody puts like the next drug and the next nanoparticle and the next tumor model. But what are really the fundamentals when it comes to, why isn't this working better as we would have expected? If we can target tumors that homogeneously, why aren't we achieving many complete cures, like, for instance, immunotherapy is nowadays doing? And other people have asked the question, so what went wrong in the design, or maybe in the conception, or maybe even the publication of nanomedicine results in the literature? How can we make this right? And that's sort of what we've been trying to do in the last couple of years. We've focused on multiple different elements. So this is just 4 different fields where I think we really need to rethink what we're doing. We need to integrate nanoparticles in combination therapies. We need to use them for immunotherapy priming, for checkpoint blockade priming, for nano-vaccination approaches. COVID really highlighted that very, very nicely. We can do different types of drugs. mRNA without a nanoparticle would never be a drug. Combinations of drugs in one nanoparticle hold a lot of promise. But what I think is the most important, and what we'll discuss mostly today, is how do we figure out what are the right patients to treat with nanoparticles? Because that is the most, if you ask me, like the biggest problem, the biggest oversight is that we're not treating the right patients with nanoparticles. If you look at the kinase inhibitors or the KRAS or some of the, even the targets that Professor Waring talked about before, people will always do a biomarker test. So you either do a circulating tumor cell test, or you stain a tumor biopsy to see whether a certain genetic mutation or a receptor target, in the case of antibody therapeutics, is present. In nanoparticles, we don't do this. So we need to figure out what are the right ways to identify the right patients. Before we move to the solutions, 2 more issues that I think for people on the call are important to consider. One of them is that among the criticism that has been received by the cancer nanomedicine tumor targeting field is that we're not efficient in how well we can target tumors. So there has been a meta-analysis by Warren Chan and colleagues a couple of years ago in which they found that in preclinical data sets, the amount of nanoparticle that accumulates in tumors is only 0.7%. Right? There are many things you can say about this number, the way how it was calculated, the fact that it's not per gram tumor tissue, which is how we normally plot it. But what I think is much more important is that this number really does not matter at all. Like really not at all, right? Because if you look at the clinic, and I hope you can read these numbers here. This is Liesbeth de Vries, a Dutch oncologist, nuclear medicine physician, and antibody specialist. They do a lot of radioactive imaging studies in patients with antibodies that are approved as products. And these antibodies in human patients achieve concentrations that you can read here, right? 0.1, 0.4, 0.3, and 0.9. So all of them are in the same range. And even though you might perceive 0.7 as bad, right? It's not bad because all of these antibodies achieve a lot of clinical benefit, patient benefit, day-to-day, quality of life benefit for patients. But that's only because they're given to patients in which they work, because we know the receptor is expressed. So how can we figure out that nanoparticles actually get to the site where we need them? The other thing is that there are reports in the literature, sometimes by people that really know their stuff, sometimes by people that don't really know their stuff, that the EPR effect or this tumor accumulation effect of nanoparticles doesn't happen in the clinic. It's only in mouse and rat models, right? So in this abstract, in a good journal, Journal of Controlled Release, people say, the EPR effect is present in rodents, not in humans. It can't be proven in the clinic. This is complete nonsense. So you can see here, like this is like a compilation of what is known in the literature. We know that in tumors, in sarcomas, in, again, Kaposi sarcoma, in the head and neck, in lung cancer, in breast cancer, we can target nanoparticles to tumors. If you do quantitative SPECT imaging, 15 out of 17 patients that were scanned in this particular study are positive for liposome accumulation, right? So that's 88%. That means it's complete nonsense to say it doesn't work. But the issue is it's very, very heterogeneous. You have values normalized to kilogram, 2%, 3%, 5%, up until 50%, 47, 32, and so on. So if you want to run a clinical trial that is maximized to be effective, you know, if you have enough patients to choose from, maybe we should only do patients that are 20% above, because then the likelihood that the drug will work or the nanoparticle drug will work is really, really high. Right? And I think that should be the starting point for how to figure out not to treat everybody, but to only select the patients where we are at least sure that the particles get there. Because if we look at this, you know, nowadays clinical trials are done blindly. That means the bottom half here, everything 5 and below, which is like half of the patients, there's not enough nanoparticle accumulating in a tumor. So even if the drug is super good, if the nanoparticle is you know, the reasons why the nanoparticle doesn't perform doesn't mean the nanoparticle is not good. Its blood perfusion is not good. Stromal penetration is not good. Maybe vascular leakiness is not high enough. We need to control the heterogeneity in physiology to find the right patients that we can treat with nanoparticles. And for that purpose, we can either use imaging or other types of biomarkers. I'm going to give you 2 examples. The first one is on imaging. We worked for many years with a company in the Netherlands from a good friend of mine that also worked back in the days at the University of Utrecht, Christiane Rijken, who started Crystal Therapeutics. I think this is one of the best nanoparticle platforms I've ever seen in terms of chemotherapy delivery to tumors. It looks pretty complex, but it can be upscaled quite easily. It can be reproduced at, you know, kilogram levels, 30, 40, 50 liter values or volumes to be used in patients. It's based on a block copolymer that self-assembles in a micelle, and this micelle is then cross-linked to fully stabilize the micelle at whatever size you desire with the drug present on the cross-linker in the micelle. And by choosing the right linker on the cross-linker, we can even control drug release kinetics, right? So this is really as good as it gets with regard to controlling, you know, nanoparticle sizing, accumulation, and so on. In tumor models, we did a couple of studies. 4T1 is a triple-negative breast cancer model in mice in BOLP-C. Syngeneic, it's by far the most preclinically used tumor model. We evaluated in these mice how well the micelles accumulate. We get up to 20% of the injected dose in tumors. On the right, you see the ex vivo imaging information. This is transillumination optics, so it's not IVIS optical imaging, it's transillumination, which is very different. It's much more accurate. We do see that we really target tumors very efficiently. And if we compare this to the clinical standard of care formulation, which is liposomal doxorubicin or liposomal pegylated liposomal chemotherapies, for liposomes, we get 10%. For micelles, we get almost double the amount, which really shows that more than 20% or so in the preclinical mouse model is sort of the max that we can achieve. With these mice, we've shown that if we load them with an imaging agent and with a drug, so Cy7 is a fluorophore, paclitaxel is a taxane drug, very hydrophobic, difficult to administer. It needs formulations like micelles to get it into mice and into humans. We can nicely see that if we treat mice for multiple weeks with the theranostic micelle that contains the drug and the imaging agent, that if we have a high accumulation of the micelles, which you see here on the right with these clouds, the tumors are actually very small. So the tumor size is reflective of the amount of the micelles that accumulate, thus more chemotherapy delivery and so on. This seems very straightforward, but this has never been really properly proven, not even in mouse models. And more importantly, even here on the left, you see that tumors where we do not have a lot of micelles accumulating, actually, they keep on growing. So the more of the nanoparticles accumulate in tumors, the more efficient the treatment is. We are quite interested in understanding how this also works in metastases. I'm not going into the details here, just giving you the context and to get a grasp on understanding what makes lesions targetable. We need to understand which features of the pathophysiology of the tumor, the vasculature, collagen, immune cells, lymph vessels, and so on. What makes lesions in the lung or in the liver or in the bone marrow, what makes them targetable for different types of nanoparticles? And we're trying to really, understand from the pathophysiological point of view, what features are the main drivers and what can we do with those features? Last example for the imaging is that we did do a study with people in Amsterdam in the Netherlands where we labeled these core cross-linked micelles with zirconium-89, long-lived PET radioisotope. They were loaded with docetaxel as a drug, and then we monitored their biodistribution in patients, multi-metastatic patients. So all of these were heavily pre-treated patients with many different types of metastases. So that is in a way a nice way to study in which metastases do the micelles accumulate. This is an example of a facial metastasis. This is the nose of the patient. This is a tumor where you see nice micelle accumulation in some parts of the tumor, but not in the whole, let's say within the whole surface area of the tumor. And actually the total level of accumulation is also not so high. This is an example of a patient with 2 metastases in the lung area where you see the spine here and the heart here. These are the lung metastases, and this is where the micelles accumulate. So here we have 10 to 12% of the injected dose within the tumor mass, nicely homogeneously distributed. If we would do a clinical trial and if we would use imaging to stratify patients, this would be an includer in a clinical trial, and this would be an excluder, right? Because it's unlikely that this patient will certainly respond well. Whereas in this case, if the drug works, if the micelle is functional, releases the drug, it's pretty likely that against these 2 lesions, then the micelle formulation will actually work. So the downside of this is that imaging is very, very accurate. It gives us highly specific information on how much of the particle, how much of the drug make it to the tumor. But imaging is not very simple, right? Imaging is complex in the sense that you need to order radioactivity for each individual patient. You need radiopharmacy. You need radio labeling tools. You need to purify the probe. Typically, patients need to be injected, and then they will be imaged at day 1 or day 4 after the injection, not immediately, because these nanoparticles circulate so long, and we don't want to image the blood pool. We want to image what is actually accumulating in the tumor. So that complicates the whole situation for imaging. And maybe it would be easier, arguably less specific, less accurate, but certainly much easier to just look at biopsies. So maybe we can stain biopsies that are available for almost all tumors, right? Some types of brain tumors are not biopsied, but the vast majority of tumors, they get a needle put into them to extract tissue to stain whether it's really a cancer or not, because that we need to know, first and foremost. And second, we want to know the proliferation rate. We want to know whether certain markers are expressed. We want to know which mutations may be present. All of that is needed for patient stratification. Maybe we can also extract information that helps us to find whether nanoparticle therapies work or not. So what we did together with AstraZeneca, quite a long project. This took us a couple of years to get done. It's enormous number of tissue features we looked at, tumor models we looked at. So we did 23 different features that in one way or the other are related to the drug delivery process, from blood vessels like density or size or distribution to how mature the vessels are, how well perfused they are, how angiogenic they are, to immune cells, to cellularity, to collagen levels. And then we actually started off in 3 different tumor models. So we took one model, A431, where we know tumor accumulation of a prototypic nanoparticle is not very good. The second model is intermediate, and the third model is sort of among the best models we know when it comes to nanoparticle tumor targeting, CT26 colon carcinoma. So these have levels that are, up to 10 to 20% per, quarter of a cube centimeter. So per gram tissue, this would be in the range of 30 to 40%. It doesn't get much better than these types of tumors, but we use them as a positive control to understand what features in the pathophysiology of the microenvironment drive accumulation. So we started with these tumor models. We stained all those 23 different features. And just as an example, this is an F480 staining. So this stains for the number of macrophages, which is in blue here. Blood vessels are in red. Macrophages are in blue. This is low accumulation. This is high accumulation. This is super high accumulation. And what you can clearly see is that both blood vessels and macrophages, they actually scale with the accumulation of the polymers. Right, so this is macrophage in the low, the medium, and the high. This is all over the different tumor models. We really see that those features are defining. And we looked at all those 23 different features. 5 of the 6 features we found to be statistically correlating with macrophages, with tumor accumulation, are related to the vasculature, and the 6th one is macrophages. So what we then ended up doing is we did a validation study, really, really big experiment with 10 different tumor models in which we had very high liposome accumulation down to very low liposome accumulation. And in those models, let me just check these things in the chat, sorry. So this is the highest, this is the lowest in terms of liposomal doxorubicin or liposomal fluorescence accumulation. We took 3 slides for each of the different tumors, and then we stained them for blood vessels and macrophages, so CD31 and F480. And then we provided them to 10 different observers, out of which 5 were clinical pathologists. And then we plotted, here in the bottom left in panel C, we plotted the number of, or the frequency of certain scores being observed. And you really see this differentiation with this cluster here and this cluster here, which are the ones that are either very low in blood vessels and macrophages and the ones that are very high in blood vessels and macrophages. And what we did is, so we gave a score, 1 is absent, 2 is low, 3 is medium, 4 is high. And then we multiplied blood vessels with macrophages to have like a maximal dynamic range. So this would be a 2 and 2 score, low and low, that would be a score 4, so here. And this is a score 16, which is like high and high, which would be here. And for this, we know that we're in this range. So we know that high blood vessels, high macrophages result in a lot of nanoparticle accumulation. And clinically, most importantly, we can identify this range of tumors that are low on blood vessels, low on macrophages, that will not accumulate nanoparticles, and that will allow us to identify patients that do not accumulate nanoparticles well enough to be likely to respond in clinical practice. So putting that in context, I think there are a couple of, for the more, people that are really into the mechanisms of tumor-targeted drug delivery, the EPR effect is kind of like, it's not outdated. It's still, you read it in papers every day, almost on a daily basis, you see new papers coming out. Some people, Warren Chan and others, have proposed things like active transport and retention, where things go through endothelial cells. So not between junctions in between, but actually through the cells. We've been trying to balance this. I believe that the majority of tumors that show high nanoparticle accumulation, which are from a clinical point of view, the ones that are most important because those are the likely responders, they still have predominantly vascular leakiness, plus a little bit of transcytosis. In tumors in which not a lot of nanoparticles accumulate, it indeed seems that transcytosis is more dominant than leakiness, but clinically, those would be the ones that we would want to exclude, right? So clinically, those are probably not the ones that are most important. Second thing here is that people have identified, again, Warren Chan and others, they're looking at these nanoparticle transport endothelial cells. We haven't been able to figure out a surface marker for pathological stainings, but if there would be a CD marker and an antibody that can stain only the endothelial cells that are responsible for the transport, then we should probably stain for those, right? Instead of for all blood vessels. And that would allow us to do even better stratification of the patients based on biopsies most likely to respond to therapies. Last but not least, I'd like to point out this Viewpoint article. It's some sort of an interview format that Nature Reviews Cancer and some other journals do. It's a cool sort of interview with 4 different people with 4 different backgrounds in which they ask questions about which tumor models, which nanoparticle types, nanodiagnostics, nanotherapeutics. One of the questions is, why so many papers, so few drugs? And you get 20 different answers, right, from 4 different people. So if you're interested in the topic, please do take a look. And with that, I've come to the end. I hope we're still in time to take some questions. Main points are that nanoparticle tumor targeting really works in patients, not only in mouse models. It's heterogeneous. We need protocols for selecting the right patients to treat. Imaging is certainly by far the best, especially using theranostic probes, but too complicated. So we may need biopsies, which are more feasible, a little bit less accurate probably. And then in a broader sense, from a tumor targeting point of view, we should really think holistically, right? So what probes make sense in what combination therapy concept? Which things combine with immunotherapy? Immunotherapy is nowadays very popular for very good reasons, because it's really helping end-stage patients to get complete cures. And we know from vaccine studies, we've known for many years, that nanoparticles target immune cells. So we can also target nanoparticles not directly to tumors, but to myeloid cells in the bloodstream, or even in the bone marrow, or into tumor-associated macrophages within the tumor. And if we make those from anti-tumor, from anti-inflammatory to pro-inflammatory, we can use those to enhance the efficacy of checkpoint inhibiting therapies and so on. So I think there are a lot of new directions that even go beyond the classical concept of nanoparticle tumor targeting. And that, together with the right type of biomarkering to find the right patients, what Professor Mike Waring showed previously, you saw these Kaplan-Meier curves nicely showing for some of the kinase inhibitors that they work, but they were actually the biomarker positive versus the biomarker negative curve, right? That's how it needs to be done, and that's what we're not doing in nanoparticle drug delivery, and that's what we should really be doing in the years to come. So with that, I'd like to give the word back to the chairperson, and I'd be happy to take any questions if there would be any. Thank you.
Thank you so much, Twan. Yeah, wonderful. Yeah, wonderful talk, and I will definitely make sure to read that viewpoint. Really interesting and great to just see that whole story across, drug discovery, drug delivery, and how we can think about how the two interact. If anybody does need to pop off, that's absolutely fine, but we can stay and take there's a couple of questions if our two speakers don't mind hanging around a little bit longer. So one question is from Antenna, which says, hello, Professor Twan, thank you for the great talk. Please tell us what the fate of the nanoparticles are after helping the drug to reach to its site of action. Can we scientifically explain what their elimination mechanism is, or are there issues of toxicity to the recipients? Thank you.
So if we're talking about the site of action, right, that will be the tumor. They would end up in tumor-associated macrophages. The majority of nanoparticles end up in phagocytes, in tumors, and also in healthy organs. If we work with biocompatible, biodegradable, lipid-like particles like liposomes, I don't think there are a lot of issues related to toxicity, also not in the liver. People always are concerned if they see biodistribution imaging studies with nanoparticles, particularly lipid-based nanoparticles, that the liver gets a lot of side effects. That's actually quite okay. The most prominent side effect is cholesterol deposition in the liver, which makes the liver a little bit more fatified. And I think it's actually an underexplored opportunity, like at least 10 years ago now. Nowadays, more and more people are looking into this, to use the fact that we know that we end up in tumor-associated macrophages towards using those macrophages as propellers of therapeutic efficacy, which would be very easy if there would be a bunch of small molecules that polarize macrophages from the, let's say, old style M2 to M1 phenotype. But unfortunately, there are no good small molecules to really do that. There are some proteins, they're difficult to load and so on. M1 to M2 is much easier. And I think in the next couple of years, we'll see a lot more efforts where people will just come up with lipid nanoparticles that then transfect the macrophages to either become CAR-M, chimeric antigen receptor expressing monocytes macrophages, depending on where you transfect them, to make those macrophages within the tumor eat the tumor cells, or in parallel to just make those macrophages more pro-inflammatory to boost the anti-tumor efficacy of immune checkpoint inhibiting antibodies.
Wonderful. Great. Thank you so much. Lots and lots of questions coming in. So I don't know if we'll be able to get through all of them, but Nenad has said, thank you for extremely informative and useful presentation. Which method would you recommend for quantifying drug uptake during drug delivery into, not on, tumor cells? So which method would you recommend for quantifying drug uptake?
Yeah. So if it's uptake into cells, the most quantitative method that we've worked with in the past is just radioisotope labeling. There you can differentiate. You can just, you can spread the cells from the supernatant. You eventually know what's in the cells, but you don't really know where it is. Many people look at fluorescence at the in vitro level, where you can see what is in the endosomes, what is in the cytoplasm, etc. But then people don't consider that fluorescent agents are oftentimes affected in terms of signal intensity with regard to whether they're intercalated in the DNA or whether they're at low pH or they're at high pH. So we can quantify, but how accurate the quantification is, we don't really know. In vivo, this is actually really difficult because properly extracting drugs from tissues, like even with internal standards, always has like an inherent range of variability and inaccuracies. What we've been using quite a lot is just standard tissue extraction with optimized protocols based on spiking with known amounts of something, and then do UPLC, like ultra high performance liquid chromatography. Mass spec for some drug molecules may work. We've been aiming with some people in Munich and in the old days with Maastricht to do imaging mass spectrometry, where you can look at the distribution, not super quantitative, but at least relatively quantitative distribution of different metabolites in tissues. But that depends a lot on how well you can ionize different compounds. So it doesn't work in our hands for doxorubicin. It does work for taxanes and some kinase inhibitors. So I think there are a multitude of methods, and depending specifically on what the person is asking, like, is it in vitro, in vivo? Is it compartmentalization within the cell, or is it more tumor versus liver? I think there's an optimal answer for each of the situations.
Great, thank you so much. Okay, maybe we have time for 2 more, let's say. Noor has said, thank you for the insightful talk. Considering the variability of the EPR effect, do you see potential for SIRS, especially with gold or silver nanoparticles, in helping characterize the tumor microenvironment or assist in patient stratification for nanomedicine-based therapies?
Yes, I think I see the value particularly in figuring out either in fundamental preclinical studies. We just had some samples shipped from a collaborative group in China where they work with gold nanoparticles, not per se for SIRS, but to just understand to which level those highly quantifiable nanoparticles can be used in similar settings where we quantify microenvironment features to predict whether a nanoparticle accumulates. Where I see clinically the most value is probably in determining surgical margins. If you do like a multi-step approach in which you, for instance, first use whole body imaging to identify roughly where the tumor margin is, and then you use something that is more accurate like MRI, like whole body could be positron emission tomography, then you can go with MRI to sort of zoom in with multi-parametric MRI to see even more clearly where the margins are. And then during surgery, you could use handheld SIRS devices or other types of, there are various types of Raman or local intraoperative devices that you can use to define what the accurate tumor margin is. And that helps the surgeon in a surgical setting to make sure that they're really cutting around the margins in the right way, provided that the theorem or the hypothesis holds up that the surgical margin is defined by inflammatory cells that take up those SIRS nanoparticles. And that combined with the fact that SIRS can be reasonably easily done in a handheld or even, let's say wide clinical setting, you don't need like super fancy equipment to do this. I think that's sort of the potential of the technology. It's potentially widely available, broadly applicable. But we're not there yet to say to which extent it really helps to improve margin detection as compared to standard surgical approaches.
Okay, great, thank you so much. And yeah, maybe let's just go for one more. All the questions will be exported so people can continue to drop them in, and then hopefully our speakers, I think most of them these are for Professor Lammers now, but yeah, if they wouldn't mind following up. But yeah, could you please comment on the increasing problem associated with PEG as the current gold standard in DDS? Oh, it's moved up. I'm referring to the rapid blood clearance of PEG-based DDS and the increasing observation of the anti-PEG antibody development in first world population by Rayner.
I think if I look at the Q&A correctly, it comes from an expert in the field that knows how to deal with potential polymer-associated issues. I think, alternatives like sarcosines or oxazolines that people have been looking at, they might, to a certain extent, be able to overcome these issues. I always challenge people from let's say the PEG alternative fields with the fact that even though PEG has these issues, there hasn't been conclusive proof that having those alternatives, one, does not result in these anti-immune responses, etc. PEG per se might have this storage disease issue. It might result in less controllable pharmacokinetics because the second dose or the third dose might not give the same pharmacokinetics as the first dose because there may be antibodies or other types of immune responses that recognize the PEG that makes it less likely that the particle will behave like we want it to behave. I don't think the evidence, particularly clinically, is available yet to show, one, that minimizing the variability in PEG pharmacokinetics, if PEG is present, is clinically meaningful in an evidence-based manner, and two, that the alternatives that might be out there are really better in an ideally double-blinded head-to-head comparison setting of controlling those issues that may result from this, right? There are many claims that some polymers are biodegradable, they're not immunorecognizable. There's some evidence for this, and some other people and papers, they disprove those points. I think particularly from a product development point of view, it's absolutely worthwhile to work in these ethylene starch and oxazolines and polysarcosines, because they provide unique IP space for PEG alternatives in a nanoparticle formulation. Whether in the end they're really, really better, I don't think we cannot say at this point in time, at least not with my, I try to really stay like neutral in terms of interpreting what I've seen. I think that the rationales are out there, but the evidence is not, for me, not convincing enough. And I think that in the future, particularly with the market filling up, we will see a couple of PEG alternatives coming up for sure in the next 5 to 10 years. Whether I know that some of the larger pharma companies, even the ones that are in the big COVID vaccine stories, they bought PEG alternative patents, but they shelved them because the PEG problem clinically is not large enough to be solved with alternatives, right? It might be an issue. There is data that in some patients it causes an allergic reaction or infusion reactions. If you want to avoid infusion reactions, you can also just slow down the infusion speed, and that already solves part of the problem. But then long-term storage, I don't think will be a problem in humans per se at any point in time. Better controllable pharmacokinetics, and making sure that the first and the second and the 10th dose will all perform the same way. That may be very relevant in specific applications where sustained control over pharmacokinetics is important. That may be cancer, that may be other things. And I think we just need more experiments and more expert panels with people very openly discussing what are the pros, what are the cons. Also, PEG is very, very well known. It's in many chocolates or shampoos. It's totally considered to be safe, whereas some of the alternatives have never really been tested in patients. So how do we deal with that issue? Pharma says, oh no, we'll just stick to PEG, at least for this point in time. So this is absolutely something that has started to come up in the past 10 years or so. And this will keep on dominating discussions and product developments in the next 10, 20 years for sure. I hope that in a way answers the question, at least gives the context.
Thank you so much. Thank you for that. And thank you. So many people have stayed on for this. So just shows how fruitful these discussions are. And like I said, terrific questions. There's more coming through. But thank you. We'll close everything there. But thank you so much to Professor Waring and Professor Lammers for taking the time.
I'm not sure if you can do that, but you can send the questions by email or something. I'm not sure if there is any function in Teams or Zoom or to just send them all.
Yes, I believe they will be exported from Zoom, and then we can send them across that way. So we can continue the discussion offline, but I think we could go probably all night. It's obviously such an important topic, and very, very amazing. So thank you everybody for being involved. And we'll take that offline and we'll close there. But thank you again.
Thank you.
All right. Take care. All right.
Bye.
Bye-bye.